Hi, my name is Reagan Bushok, and I'm a sophomore studying biological engineering. What if I told you that the human microbiome is crucial in understanding cell signaling pathways in our response to disease? A microbiome is a community of trillions of microorganisms, including both symbiotic and harmful bacteria, viruses, etc., that exist in various environments in our body. A person's microbiome has a large influence on our health, including susceptibility to disease and infection. The Brubaker Lab, under Dr. Douglas Brubaker within the Biomedical Engineering Department, develops systems biology approaches to model host microbiome interactions and identify novel therapeutic strategies for complex inflammatory diseases. The lab focuses on research areas including Alzheimer's, IBD, and bacterial vaginosis. I am a part of the urogenital microbiome group, where we focus on understanding the vaginal microbiome affected by bacterial vaginosis, in which vaginal inflammation is caused by the overgrowth of bacteria naturally found in the vagina. Our current studies involve researching the relationships between metabolites and proteins involved in bacterial vaginosis to see how vaginal epithelial cells respond. Using this information, we are currently developing a computational model to predict vaginal epithelial cell responses to microbial metabolites and establishing an experimental system, in this case, a vaginal cell line, to test hypotheses extracted from the model. I am currently working on identifying the most prevalent metabolites and proteins in clinical sets of data of patients with bacterial vaginosis and analyzing which relationships between these metabolites and proteins have the strongest correlation. Within this heat map that I created recently, the 4 bacterial strains are on the right-hand side, and all 178 metabolites are listed on the bottom. Red means that the metabolites are very prominent, whereas blue means they are severely lacking. If we know what to target and why this signaling is happening, we can begin to engineer a solution. One avenue in current discussion is the creation of living engineered bacterial therapeutics, seen on the bottom right, in which bacteria are engineered to process signals within the body and produce a calculated response. This is done by creating a gene circuit in which an assembly of biological parts that code for RNA, proteins, etc., allow bacteria to respond to specific signals and interact with their environment to produce an output. In this case, sensing bacterial vaginosis and outputting therapeutics. Though we are far from completing this task, members of our lab are currently undergoing training and collaboration through Dr. Leopold Green within the Green Lab to design and construct unique transcription units and gene circuits in preparation for this step down the road. Not only does our research target the how and why of cellular mechanisms involved with bacterial vaginosis, but we are actively preparing to engineer a solution to improve women's health and take one step closer to understanding the human microbiome. Thank you.